Thanks again, you guys, for coming in today. I'm not going to try to muddle my way through your credentials. I'll let you introduce yourselves because I don't know that I would get them right. So I'm calling you Dr. B because I know I can't screw that up, but please give us your full title and credentials and everything.
Yeah. So my name is Dr. Eric Birgbauer. I'm an associate professor at Winthrop University. Received my PhD in cell biology from the Massachusetts Institute of Technology. Also
locally known as MIT.
Yeah.
Very nice. And how long have you been at Winthrop?
Almost 15 years now.
Wow. And Jeff, tell us about your background.
I grew up in Fort Mill, South Carolina, and went to Winthrop. I did my undergraduate there and a bachelor's in biology, and now I'm there to get my master's.
Nice. And what's your master's in?
It'll be in biology with, I guess, a concentration in cell.
Nice. So we were just talking kind of right before we realized that we were live. So I'm a Winthrop grad, and I studied political science, which, obviously there's some tension of, from people that are in hard sciences versus the liberal arts when some people laugh when we say political science. But honestly, I've been in and around Winthrop since 2002, and I was completely unaware of the level of research that goes on at Winthrop, and how researchers such as yourselves are really moving the needle forward and taking great strides in what is cutting-edge science and technology. And when I found out about what you all were working on, I said, I have to learn more. I have to learn, about how this goes on at Winthrop, what it is y'all are working on, and what does that mean to the world. So can you tell me a little bit about the research that you have going on? And I guess we'll just start there.
Okay, so I have a research lab there at Winthrop University, and I'm studying what I call developmental neurobiology. I'm trying to understand how the brain forms. And specifically, I'm looking at how the connection from the eye to the brain gets established. That's through the optic nerve. And initially, the nerve starts off at your eye, and during development, it grows, and it has to connect up to the right place in the brain. And so trying to understand the molecules that are involved in the specific guidance of this nerve up to the proper place in the brain.
Okay. So when does this happen? So you're talking about during
this happens during fetal development.
Okay.
Yep. So during fetal development, for ethical reasons, we don't work on humans. We actually use chicken embryos because they have a very similar early development to human. But they're nice and easy to obtain in what I call little packages called eggs. We can access them much easier. We can do experiments. We can even take little pieces of the early developing eye out and culture it in a culture dish. The nerves will grow out from that piece, and we could see the effects of different compounds or different chemicals or different axon guidance cues on the nerve of the growth of those nerve cells.
So what do you hope to obtain from this?
Yeah. So during development, the nerve grows and can connect up. But if the nerve is injured, it does not regrow. So CNS nerves do not regenerate. And so the question is, well, what is different about the developing nerve versus the adult nerve? And why doesn't it regenerate? And if we understand more about what happens during development, we might be able to induce regeneration of the adult nerve.
So what are you finding so far?
So we're looking at a very specific molecule called LPA, or lysophosphatidic acid, that so far seems to be inhibiting the growth of this nerve. And so we're trying to find out, does this inhibition actually affect it during its normal development and growth? And what in the nerve cell is actually responding to this molecule? And so trying to look for the specific receptor that is in the nerve cell that causes it to respond to this molecule.
So there's a few things going on here in my mind. So you have the fact that this nerve grows at all, which is interesting because it's developmental versus an adult nerve. And also it seems to know where to go.
Yes.
So you're studying both of those things, right? Not just the fact that it does grow during this stage, but also the fact that this particular nerve seems to always grow to the same place nearly without fail every single time. So is there a connection between the fact that it is growing, or what's allowing it to continue to grow, and the direction of its growth, and that it seems to do so with such precision?
So potentially a connection between that, but there's also a separation. So there's things that just cause it to growth, and some of that are specific genes that are expressed that cause a nerve to grow. And then it'll just sort of keep growing in one direction. And then the question is, what causes it to change direction and basically turn and maneuver up to the right spot? So we're a little bit more focused on that second question of what actually causes it to get there. And presumably there are things that cause it to be attracted to one direction, or things that would repulse it or cause it from going to the wrong direction.
So if you can figure this out, it sounds to me as if you'd almost be able to reverse engineer the process and grow and steer a nerve to be able to go where you want it to go. Is that the idea? Is that the hope?
That would be the hope. And so again, there's sort of, as you mentioned, two factors. There's the factor of an adult nerve not wanting to grow as much as an embryonic nerve. There's also the question of it getting to the right place. And a lot of research is being done on trying to get it to grow. But importantly, even if we get it to grow, if it doesn't grow to the right place, it's not going to help.
It's not going to do any good. Exactly. So the hope is that there's hope for maybe spinal cord injuries. That's the first thing that comes to my mind. What's the broader universe of potential applications?
So again, spinal cord injury is one big area, but also damage to the optic nerve, the nerve that connects the eye to the brain. Glaucoma will often cause damage to these cells in the eye, the retinal ganglion cells that set up this nerve. And so many causes of loss of vision could be due to basically damage to this nerve. Glaucoma being one example, injury or another thing as well.
How long have you been studying this?
So I've been studying it since my time at Winthrop for about 15 years, and then some things before that for about another 5 or 6 years of other research before I moved to Winthrop.
And you said you've been at Winthrop
about 15.
Is that obviously it's a very challenging problem. What obstacles, 15 years sounds like a long time to be researching anything, but it's not my world. What obstacles have you encountered throughout that time, and how have you overcome those?
So obstacles, I'm going to say, have sort of three aspects. One is just my time at Winthrop University. I'm spending a lot of time teaching, working with students who are taking classes. We don't have time to full-time devote to doing the research. So it's doing research while teaching, students while taking classes. And so that's obviously been one aspect that doesn't get me to move as fast as I would like.
Definitely take up a lot of your time.
Yes. Other thing is technical aspects. So one of the things we're looking at is trying to find out what's the receptor molecule. There's a variety of receptor molecules for this molecule OPA that have been discovered, and so which one? And so we've looked at some, did some previous work that suggests that there's 6 different receptors, that receptors 1, 2, and 3 are not the ones involved. Receptor 5 doesn't seem to be expressed in the eye. We're not right now hypothesizing what's called receptor number 4 is the one that's involved. And so we tried one technique that's a previous technique called RNA interference to try to remove that receptor from the developing nerve. That's an older technique. It didn't work as well as we expected. We never actually got it to reduce the expression of this receptor. So recently we started to try to use the CRISPR technology to see if we can essentially mutate that receptor in the developing nerve and see what happens.
CRISPR.
Yes.
There's my next question. So tell me about CRISPR.
So CRISPR is a new technology that just came out a few years ago. It was originally discovered in bacteria as a mechanism for bacteria to try to fight off viruses. And so bacteria get viruses just like we do. And so if a bacterial cell gets a virus, the virus injects its DNA into the cell and will basically try to take over the cell. If the bacteria somehow survives this viral infection, it takes little pieces of that virus DNA and stores it in this CRISPR array, which is sort of the bacterial cell's equivalent of memory. And so if that virus ever infects it again, it recognizes it and it activates an enzyme called Cas9 that will cut that viral DNA and therefore make the virus inactive.
Wow.
The cool thing about this is if you take the sort of template that this bacteria uses, but you insert information of your own and take that Cas9 enzyme and put that into another cell like a chicken cell or a mammalian cell or a human cell, it will do the same thing. It will find any sort of DNA that matches the template you put in, and it will cut it. What happens in human cells is if it finds that DNA matches a gene that's normally there, it will cut that gene. Your body will then, or your cell will try to repair it. And so usually what it does, if there's a cut gene like that, it doesn't exactly know how it's supposed to go back, so it'll just sort of try to squeeze it back. And when it tries to squeeze it back, it will usually make a deletion or sometimes add some additional DNA, and so it won't be a perfect match. And when it does that, it creates a mutation. And so that's been used technologically to, create mutations at will in a gene that you're interested in.
So if you understand, if you have the genome, you can create a mutation to basically do designer genes. You can basically make it anything you want it to be, I guess.
Yeah, so if you have the technology to know or the knowledge of what it is you want to do, you can essentially do almost anything you want. And so that's the idea of designer
anything
genes.
you want is where things get really interesting, right?
Yes.
So, because in my mind, I immediately go to like comic books and where you think of like, Captain America serum or something, and you can do whatever it is you want to do. You can make, you can change someone however they want. What are the limitations though? So, I mean, that's what CRISPR is, but what isn't it? And, I've encountered the phrase before, like it's something that I'm somewhat aware of, but I don't really understand where the limits are.
So the first major limitation is our knowledge. Mm-hmm. We don't know how to make a Captain America. So even if we could design their genes perfectly, we don't know what genes those should be.
Ah, okay. So in other words, we're less limited by what it is than just our understanding and knowledge in general, or of that in particular.
Yeah, so a lot of it is, we can design different things, but what we want to design depends on having to know what those blueprints should be. Mm-hmm. Even something as simple as eye color is involved with a multitude of different genes. And so it's hard to get exactly the right eye color you wanted, even if you wanted to do that and could do it.
So is that really the horizon of limitation then, is just understanding genetics and just getting a little bit better at genetics in general? And so, because it sounds almost as if you're saying if we have a more complete picture of just genetics, and I'm throwing around words that I barely know what they are, but if we have a more complete understanding of just how genes work, then there's no reason why we shouldn't be able to change any genes, basically.
So theoretically, yes.
Okay.
There gets to be a couple of things that get in between theoretic and reality. Mm-hmm. There's ethical issues. Mm-hmm. Should we change things, especially to make, let's say, designer babies?
Yeah.
There's also the issue that theoretically we can change any gene we want. The problem with this technology is it works 99% of the time. Mm-hmm. What happens with the 1%?
Yeah.
Especially because the human genome is billions of base pairs long. And so you can target it to this one region, change the gene you want, but maybe accidentally it'll change another gene in the genome, what we call off-target effects. And if it does that, then we're opening a can of worms that we don't know what's going to be the effect. Often these mutations can lead to cancer, something we don't want to do.
Yeah. Where does the 1% error come in? Is that laboratory error, or is that just by the way CRISPR works?
As far as we know, it's due to basically the enzyme. The enzyme, and it's probably actually more accurate than 99%. It's like 99.9, but still it targets this with great accuracy. But because it's an enzyme that can cut DNA, if it sometimes makes a mistake and cuts someplace else, then again, you could have a mutation in a gene you don't want to.
So you mentioned that this comes from bacteria.
Yeah.
Is there a specific kind of bacteria, or is this a mechanism that's common in nearly all types of bacteria?
So this particular one was developed from a particular bacteria, and I can't remember the exact one right now.
Streptococcus.
Was it Streptococcus?
Streptococcus.
Yeah. But other bacteria have been found to have similar mechanisms, either a similar Cas9 type of protein, or it could be a similar but different type of protein that seems to have the same property.
So I guess there's a long-term view and a short-term view. Long-term view is we really don't know what we'll be able to do with this. There's no telling. But in the short term, are there more immediate applications of CRISPR in general in what other fields are people utilizing this research or utilizing the CRISPR mechanism, and in what ways is this technology furthering those other fields?
So one of the major ways it's being used is being able to specifically mutate a gene that you're interested in studying. And so if you think this gene, let's say, may be involved in Alzheimer's disease, and if you could mutate it in some sort of cell culture system or some animal model like a mouse model, then you could see, well, what effect does it have about mutating this gene? Would the mice or the cell system get properties similar to what you'd expect in Alzheimer's disease or any other particular disease or aspect you're interested in? Like for us, there's this particular receptor gene we think might be involved in guiding this nerve. Well, if we mutate it, will it still be guided properly or not?
So then you can see what the differences are when you post mutation in another chicken.
Yeah.
So why you said chickens are very easy. It's funny because you always hear about lab experiments with mice or rats or something.
Yeah.
It's the first I've ever heard with chickens. So there's enough of there are enough similarities between the chicken development and their brain and humans in order to be able to make comparisons and actually have usable research even with chickens.
Yeah, so there's enough similarities. They're not obviously humans or that close, but the early development, there's a lot of similarities. And chicken hasn't been used much for adult research. People haven't really done chicken farms. But the early embryonic development has been done a lot. And one of the big advantages is mice, rodents, just like humans, all the fetal development occurs in utero, which is a lot harder to access and a lot harder to do manipulations in. Chicken embryos occur in eggs, and so it's a lot easier to access them. And there's even ways to open up the egg and take the embryo out and culture it and do experiments in this culture system.
Interesting. And so you're working in the labs, right? So you're technically a grad assistant right now working because you're working on your master's?
I am, yes.
Is that right?
Assistant, yes.
So tell me about your role and how y'all work together.
So Dr. B is my mentor. Basically, I'm doing research in his lab on a PRG, which is a possible receptor for LPA, but scientists aren't really sure exactly what it does yet. Like the G protein receptors you mentioned earlier, there's a class of 5 of them. I think there were 6 of the G protein couple receptors. And the PRG receptors are found in adult brains, and they may play a role in, I guess, neuronal guidance in the hippocampus specifically. So plasticity-related gene is what it stands for, so a role in memory and learning. And so basically we're trying to discover whether PRG is going to respond to LPA in some way, shape, or form.
Now, what do those things stand for? What is PRG?
PRG stands for, plasticity-related gene.
Okay.
And so the plasticity-related gene codes for an mRNA. And this mRNA makes a protein. This protein sets on the outside of the neuronal cells usually found in the hippocampus specifically, or in the cortex of mammalians, humans, and I guess the thalamus also. And scientists are interested in what this receptor is doing on the front of, or the outside of the cell. Is it responding to LPA in a way that makes it grow, or makes synaptic connections, or disassemble those synaptic connections?
So again, for me, the question, the edges of science are always are so technical and specific, and for me, it's difficult to understand the why, and what do we hope to learn from this, and how does this research move the ball forward on a macro level? So explain that to me.
So on a macro level, a lot of people are interested in diseases like Alzheimer's or ALS, or any nervous system disease. Right now, we don't necessarily have cures for those diseases, and so a lot of people end up suffering from them. And so one way to understand those diseases is to know, we know they ultimately result in some type of damage to our central nervous system. And so we want to know how to stop this damage and or revert this damage.
So there's been how many generations of scientists researching, Alzheimer's and ALS, and kind of all persistently attacking the same idea and theory for a long time, it seems like.
Alzheimer's research started in the early 1900s or late 1900s.
Have they been on the amyloid hypothesis that whole time?
A good bit of it has centered around the amyloid hypothesis, but it's starting to change in more recent years.
Because it sounds like you're on a different track than the amyloid hypothesis. Can you talk a little bit about that?
So, as far as the amyloid hypothesis goes, the theory is that amyloid beta results in Alzheimer's disease. Amyloid beta is a protein that is cleaved from a natural protein found within your membrane in your nervous system. And so the amyloid hypothesis theorizes that this protein is cleaved excessively as you age, and this buildup of this protein causes cognitive effects that are seen in Alzheimer's, the neurodegeneration. It builds up it builds up nerve tangles around neurons and causes them to, degenerate eventually. And so that's the basis of the amyloid hypothesis. And so now a lot of research was put towards that end, saying, okay, this is kind of like a causation type deal where we're going to follow this line of research, and they only considered that as the main possibility, that amyloid beta is going to result in Alzheimer's disease. Now,
and not the inverse. Right.
Well, now there are multiple perspectives, and so we found that everybody that gets Alzheimer's disease doesn't have high levels of amyloid beta. And so there's no direct causality from the amyloid beta, but it plays a role in Alzheimer's disease. And in pretty much every theory there is currently, amyloid beta has some sort of role in Alzheimer's disease. But now scientists are moving away from just the amyloid hypothesis. There are other things affecting us that could cause Alzheimer's disease. Pathological insults from herpes virus is one. Tau abnormalities, which is another gene or protein. And so that's why, science is mainly moving away from the amyloid hypothesis per se, but amyloid is always going to play some kind of key role in Alzheimer's disease and how it's researched, because it's there and it has a strong linkage, but it's not a direct causation.
So it sounds like for a long time everyone was hoping for the single bullet. We can solve it with this one thing. But you're saying it may actually, or these type of neurological diseases may come from a confluence of different factors, all of which could play a role. If all that is true, what does that mean for the pharmaceutical world? What does that mean for anyone hoping to find something that could help prevent these diseases?
Well, there's never going to be one pill that you have to take to get rid of it. It's probably always going to be some series of treatments to cure these diseases, or may not even cure them, prevent them, or lessen the effects that we feel from them.
So it's always going to be about mindful. You'll always have to be mindful of something. It's not a single pill that you'll be able to take. It sounds like it's almost a lifestyle thing. If it's a confluence of all those factors, then that's pretty profound. That requires very diligent effort from anyone trying to prevent those things, right?
Definitely.
So as the professor kind of guiding this research, I guess, is this something that you're working together on?
Yeah. So I guess I'm his mentor, so I'm advising him. He was very interested in these PRG genes, again, that were initially discovered because they're potentially involved in learning and memory. Mm-hmm. But there seems to be this association with this molecule that we're looking at, LPA, that we think guides the nerve. And so the question is, could these be involved in guiding the nerve?
Yeah.
And so that's sort of a new direction of our research that Jeffrey's heading up.
Wow.
And helping us to see, could this gene that we understand something about in learning and memory be involved in nerve guidance?
It's fascinating to me to hear that this sort of thing is happening in our backyard in little old Rock Hill, South Carolina. And if a university like Winthrop is able to pull off such, and these sound like pretty big leaps in the field, to be able to move forward with this research, I'm surprised. And as I've mentioned, as someone who's in and around the Winthrop community a great deal, and I suppose it's my fault, but to only really just be hearing about this sort of thing, is it unique for me to be somewhat surprised, or is that something that you encounter? Like if you're at a cocktail party and you tell somebody what it is you're doing, like what's the typical reaction?
And I think it is not unique to you. I think one thing we haven't done as a good job as a university is getting the word out that our students and we are involved in cutting-edge scientific research.
Yeah.
I think that's something that's developed over the last 15 to 20 years. I know when I started at Winthrop, it was part of a program called the South Carolina Embry Program that is to help develop biomedical research at some of these non-traditional research schools like Winthrop University. And Winthrop University has been one of the pioneers with this Embry grant that's for South Carolina, and has been basically growing a lot in their research capabilities through that. We have a lot of students working and a number of faculty doing biomedical research and cutting edge in their field.
So you mentioned the grants. Is that where the bulk of the funding for this type of research comes from?
Yeah.
Some outside grants?
Yeah. So
where are the sources for most of those grants?
So most of it is the National Institute of Health, NIH, provides huge funding throughout the United States for biomedical research. There's also the National Science Foundation, NSF, that provides funds for science research. And then sometimes there's other grants, although at Winthrop, the majority of our funding has been coming from NIH.
So I think if you're perhaps the typical high school student interested in the sciences, someone may tell you at some point that if you want to do this level of research, then you need to try to get into a research institution of some kind. I imagine that's an obstacle that you may encounter with potential applicants. Are you finding that students are surprised and open to coming to Winthrop once they hear about the type of research that's going on there? In other words, is this a recruiting tool for you when you're working on something like this?
Yes, it is a recruiting tool because so many college students, especially if they're thinking of going to graduate school or even medical school, would love to get involved with research.
Yeah.
And we don't have quite the amount and level of research as a primary research institution. But one thing we do have is our undergrads can get involved with this research. And so I have, 3 undergraduate students in my lab and a master's student who are working on it. At the big research institutes, a lot of it is PhD students or postdoctoral fellows that are doing the research. And undergrads sometimes find it a little harder to get involved because there's these other students involved.
So they're getting research, I guess, 4, 6, 8 years ahead of schedule than they would otherwise be getting if they were on that track.
Potentially, yeah. Maybe about 3 or 4. Depends on when they do it. They usually don't start as freshmen either. Take their initial science classes and then, sophomore, junior lab can start getting involved with research.
So you mentioned your background at MIT. MIT is a long way from Rock Hill. How did you end up at Winthrop?
That was a long story. So I guess for me, originally I'm from California, but did my undergrad at UC Berkeley, and then actually moved across the country to MIT. Did my PhD work there, and then in the sciences, usually you do what's known as a postdoctoral fellowship, which is sort of like a further fellowship or internship, equivalent to like residency in medical field. And so I actually went and did a postdoctoral fellowship first at Caltech and then at UC San Francisco, and in sort of neurobiology, getting more research experience. And then actually from there, I was interested in the pharmaceutical industry. And so I was working with Merck in their research labs. They had a research lab down in San Diego at that time. And so I was working with them for a while there, and then they had a reorganization, and they ended up closing that site. And so I moved on, but I decided I wanted to focus, instead of primarily on research, on something where I'd also be doing teaching. So I started developing my own research program in collaboration with a scientist at the Scripps Research Institute, started getting some teaching experience, and then got the position at Winthrop University, where I'm now teaching, but also doing research with students.
And I think that's a key distinguishing factor with a school like Winthrop, because there's plenty of schools where you could do research. There's plenty of schools where you could teach. And if you happen to actually like teaching, it's kind of hard to have the time or be able to do that at a major research institution. Is that one of the things that attracted you to Winthrop in the first place, was being able to spend time with students and actually teach in the classroom in addition to the research?
Yes, definitely. I really wanted to work and help basically bring up the next generation of students, scientists, medical professionals, wherever they are going to be. And so I really wanted to teach, and that's why I was looking at places like Winthrop. But again, I say I have my research experience and wanted to continue doing that. And I think it's also a great opportunity for students to learn what science is really like.
Yeah. And learn what it's like, not just in the lab, but I mean, you have to, I imagine, write all the grants to get the funding. You have to, work with any research limitation or resource limitations that there may be. But yet somehow, these students are getting this amazing experience. Like, you're being able to teach, you're also being able to conduct just fascinating research at the same time. So it seems like a pretty awesome situation, to be honest with you, from the outside. So, I mean, everything, the grass is always greenest on the other side, I know. But, it's pretty great to see such awesome research going on just right here. I want to get back to the CRISPR stuff a little bit, because I have so many questions about that. First of all, we kind of skipped right over it. What does CRISPR even stand for?
Clustered regular interspace short palindromic repeats.
Nice.
Yes. AKA CRISPR.
AKA CRISPR. Now I see why they call it CRISPR. It's so much easier. So if someone wanted to engage in I guess could I call it gene editing? Is that
Yeah.
Okay. If someone wanted to engage in that prior to the development of the CRISPR mechanism for these purposes, how did that occur before, and how is CRISPR an improvement on whatever that process was?
So there are a couple of different ways they had done it. There were certain proteins that could edit genes, but they were fairly complex to work with. A more common way was to basically create a gene that matched the full gene you wanted to make. So if you wanted to change a gene, you'd create a gene that would match it except for a little region that you'd want to change. And then you would inject that into embryonic stem cells. You would select any that happened to do a recombination between the particular gene you put in and the normal gene. And that would be like a 1 in a million chance. And so you'd find those that actually had done this recombination. You would select them. You could then grow them up and then implant them into, let's say if you're doing a mouse, into the early embryo, the recently fertilized egg of a mouse, and implant that into an animal. The advantage about CRISPR is frequency of this is at a much higher frequency. It's not like 1 in a million. You don't need to clone the whole gene. You just need to find the particular region of interest that you want to mutate, and you can do it with a short region. And it's, again, much higher efficiency.
Mm-hmm. So this is a vast improvement.
Yeah.
When was CRISPR first used intentionally like that? Like, how far back does this go?
1993.
Okay, yeah, I forgot the exact year.
Yeah. Okay, so we're still new in our understanding of it. I mean, that's not
when
too long ago.
CRISPR was used. I think there were 2 other gene editing tools similar to CRISPR that used nucleases, but they were hard to produce and really expensive. I think it was almost like $30 per set.
Mm-hmm.
And so when CRISPR came along in 1993, that's when it was discovered. I don't think it was actually used until the early 2000s.
Yeah, well, I think, I mean, after it was initially discovered, it was fairly shortly after that they realized the potential. So I think it was like 2 or 3 years after discovery that they started to realize that, wow. And then they started to be able to apply it in mammalian systems and realize that could use it for gene editing.
So in my mind, when you just mentioned they started to realize the potential, to me it seems almost limitless. And I don't know how accurate that is. That's why I was so curious as to what it isn't, because it almost seems like nothing short of miraculous to me that there's this potential to be able to edit genes so easily, almost like changing your tires. It seems. I know there's more to it than that, but in terms of, it just seems like there's so much potential there. And you mentioned earlier the ethics of it all. Where do the boundaries of our ethics and our own limitations on our use of it, and I guess to what degree do ethics get in the way of advancement of the research? And I guess it's the question is also the inverse of that. So in an environment where those ethics were less of a concern, is there a chance of greater advancement in the research?
Yeah. So the major ethical issue is basically, I'll say, creating designer babies, changing the genome of an individual that would basically be inherited.
Mm-hmm.
Because now it's not only affecting that individual, but it'll affect all their offspring or children.
Yeah.
And again, it's that aspect of, okay, are we playing God? Do we want to change this? Especially because we don't always know all the implications.
Mm-hmm.
I guess in China a few years ago, John Quay, I think I'm pronouncing his name correctly, tried to basically mutate this gene CCR5 that is a receptor for the infection of the AIDS virus, HIV.
Mm-hmm.
And there was some evidence that that people with this certain mutation in it were resistant to the virus. And so the idea was, okay, to create that for these 2 children. Couple of issues. When he made this mutation, it doesn't make exactly what you want. It will make a mutation, but it wasn't the exact same mutation. Would that new mutation confer resistance or not? Unknown.
Mm.
This gene normally has a function in cells. That's why most of us are not mutated in it. What's its normal function? Could you be hindering these children and making them maybe more susceptible to immune deficiency or something like that?
Mm-hmm.
So that's another big issue with making changes that are permanent.
It sounds like he made those changes without answers to some fundamental questions that existed around whether or not he should have. And I think, so I recall that story, and I think that might have been my first exposure, because there was some news media coverage when that occurred, and it's like, hey, Chinese scientists, create some mutation using CRISPR, and my question was, what in the world is CRISPR? Like, how can you just do that? And I think for a lot of folks, that was their first exposure to it, which is unfortunate because in a lot of ways that was a negative exposure to it. In other words, it wasn't, we just solved Alzheimer's, or we just solved, nerve growth, or it wasn't something like that. It was, should we have been doing this? It was an advancement, or if you want to call it that, it was an experiment that involved the outer boundaries of CRISPR ethics, as I think of one way to put it. As you talk to people about your research and the fact that you're using CRISPR, do you find that there's a positive or a negative response, or mostly neutral?
I found mostly curiosity, honestly,
Yeah.
in what CRISPR is.
Yeah.
And I know there was a huge negative backlash among the scientific community about that, but as far as the lay non-scientific community, I don't know if
Maybe they didn't really
conveyed
understand those questions that you just
severity.
referred to. Yeah, like why that was possibly in a gray area of ethics.
And specifically in the United States, CRISPR or genomic babies or anything with the genome's considered a drug under the FDA.
Okay.
So it's illegal to edit or distribute drugs without FDA approval. And the FDA hasn't approved genome editing. So you can't, at least in the United States, do any type of genome editing on humans. You can get grants to do them on non-human mammals or lower invertebrates, but you usually have to submit, grant approvals for the non-human mammalian animals.
Yeah. Although there are some clinical trials going forth trying to use CRISPR not to make babies, but on adults. There's one clinical trial going on trying to correct basically a gene that's mutated that causes blindness, and to try to correct that gene. And so again, here they're going to put the CRISPR into the eye, not into the whole person. So it's a very limited trial. And again, obviously, they've submitted this to FDA. There's approval to go on and at least try this.
So with any technological advancement that requires a robust ethics around, like nuclear proliferation, for instance, are you concerned at all about CRISPR proliferation? Is there any concern about, as our understanding of CRISPR advances, as it becomes accessible in other areas, like outside of systems where they have legal limitations on its use, or even within those systems with people who just don't care about legal boundaries? What are some of the concerns, or are there concerns about its more widespread use in research?
Yes, there are definitely concerns. And I think the situation with the Chinese scientists basically exploded those concerns, because here was somebody who was doing this research before I don't think scientists really even thought we knew enough about it, pushing the boundaries of ethics, getting around their own government controls, sort of doing it on his own. And the fact that the technology is readily available is a concern, because it's not hard to do it. It's just, are you doing it well enough? Are you doing it with enough understanding? And and then the whole ethical issue of, should you be changing a person completely? So
you mentioned, its use in changing adults, so not just at the embryo level, but, full-grown adults. Also implicit in the idea of a full-grown human is the idea of capacity and the ability to make decisions. Is there as much ethical constraint outside of the FDA requirements, but let's just talk purely ethics and not legality. In your mind, what are the ethics around an adult who may understand the risks? And you mentioned the 1% error or whatever the error may be. What in your mind are some of the ethics around a fully formed adult with capacity making those types of decisions?
Obviously, you've hit on the major thing, consent. And consent with knowledge. In other words, does the potential patient understand, at least in principle, what's going on, what the potential risks are, that any scientist or doctor working with them would be able to explain the risks, being able to have the person decide whether or not they want to participate in this trial. Because, it's not completely risk-free, and there's also the whole issue of, will it even work? It should work. Are we going to be able to get that correction done properly? It doesn't happen in every cell. It's not automatic. It will happen most of the time, but...
How far are we from the day when you can just walk into your local clinic and say, I want blue eyes, or I want blonde hair, whatever you may want? Like, how far are we from that?
I would say we're still a ways away. And again, it comes down to, obviously, will the local clinic ethically want to do that? But more importantly, do we understand what would cause blue eyes or brown hair or something like that? And then even doing it's a complicated procedure of trying to edit the genome of essentially a fertilized egg. And so it'd be in vitro fertilization, taking those eggs, trying to do CRISPR mutation on them. And then one of the questions, okay, first of all, if you want to change a gene, we all have 2 copies of our genes, one from the mother, one from the father. You'd have to edit both of them, which isn't always obvious and easy to do. And then you'd have to determine, okay, which one of these have a gene change in them? And you'd probably want to do that before the child grows up, because then it's sort of pointless. But obviously, if you have a fertilized egg and you take all the DNA out of it, you don't have a fertilized egg anymore.
Yeah, interesting. You're doing a lot of research around neurological diseases. You're doing this research along, the lines of, nerve growth and the implications what that may be. To me, one of the common themes is just human aging and how aging affects our biology. Where are the applications of CRISPR and your fields of research on the larger field of human aging in general? Can you kind of put it in the context of those things for me? Does my question make sense?
Yeah, it's a very complex question. To start with, there are a lot of processes that go on in aging. Many of them we don't understand, and especially don't understand the implications. There's a lot of things with, damage that goes on with time. How much of that damage is able to be repaired and corrected, or how much is actually going to have some permanent disability? Even something like Alzheimer's disease. It's a prevalent disease, and many of the people get it, but there's many, many people that don't. And more people that don't get Alzheimer's than do. And so what's the difference between them? And we don't know yet.
Yeah. Now, is that on a common lifespan? So I know your odds go up tremendously as you live longer, right? So over a long enough timeline.
I think it's a 1 in 9 over the age of 65.
Okay. And at what point, in other words, is there an exponential curve at some point where it says over the age of 80 or 90 or 100, you're more likely than not to have some type of neurological disease?
Yeah, I mean, I think it gets up to like over 85, about 1 in 3 or something.
Okay.
But at the same time, then there's 2 in 3 that don't. And there are many people who, they can live to be 100 and still be cognitively well aware, have no particular symptoms of Alzheimer's. I think everybody to some degree gets a little cognitively impaired, little forgetfulness and stuff. But Alzheimer's is a much more severe situation and is categorized by not only the level of cognitive decline, but also, again, these specific symptoms that are seen in the brain with this amyloid plaques and these tangles.
So you mentioned a minute ago the loss of information over time.
Yeah.
I recently tried to read a book, although perhaps if you're familiar with it, you actually may help me understand it, about the information theory of aging. Are you familiar with that? It's by a Dr. David Sinclair. Really interesting, but that was what I gathered from the book was basically, how that loss of information over time in cellular reproduction causes humans to age. So anyway, it was interesting read.
Okay, yeah, I'm not familiar with that.
But is that what were you referencing? Because it sounded pretty similar to what he was talking about.
So again, there's a lot of things that are going on in aging. There's information in your DNA. There's a chance of mutations with time. Mm-hmm. And so again, you start off with whatever information, but if there's mutations, you might not have all of that information going on. Your cells are dividing, depending on which type of cell, fairly rapidly, replacing a number of different cells at different times. Mm-hmm. Brain not dividing as much, but it's still being exposed to all sorts of chemicals, different sorts of things in the environment that could cause errors in reading DNA.
Where our worlds may sort of intersect. So obviously I see a lot of injured people. And whether it's, spinal cord injuries, or that's obviously one big area, but the other one is brain injuries. Is there an application from, like you mentioned, if we could figure out what caused these nerve cells to grow in the direction, do you think that there's also an application within the brain, with neurons specifically?
Yeah, so the spinal cord is just part of our nervous system.
Yeah.
It's used to study because obviously the effects are very dramatic. Mm-hmm. It's also a fairly isolated nerve. Your brain has trillions of nerves. And so the thing is, if there's damage there, well, what all nerves are being damaged? So it's a little harder to study. But if we could understand how nerves grow, let's say in the spinal cord or the optic nerve, which are sort of separate nerves, and if with time we can understand the different nerves and what they're doing within the brain, maybe after brain injury, there would be a way to reestablish them.
Yeah. And that type of research that provides a lot of hope to a lot of people, because we're told that, when you suffer a brain injury, it's basically permanent. Like, this is just something you're going to have to live with. You're going to have to live with impaired cognition for the rest of your life. And, for some of the cases we see, when you're talking about a child, or that's the rest of their life. That's a very long time. That hope represents a fundamental paradigm shift in how we deal and treat people with injuries. Not just, nerve injuries, brain injuries, and I guess they're all the same thing, but that completely changes the idea of medicine in an entire field of what we can possibly do. When I think about these things, I think about timelines. What does this mean 10 years from now, 20 years from now, a generation from now? Do you think it's conceivable that within a generation or two, those types of injuries are no longer seen as permanent?
I think there's a chance within maybe, 20 years or so that there will be some way to have therapy for, let's say, optic nerve damage or spinal cord damage. It may not be complete restoration of function, but it may be significant restoration of function. Mm-hmm. In other words, a person, let's say, may not walk completely normally, but they may be able to walk.
Yeah.
Which would be
That's
a big improvement.
Difference. And you ask anyone who's in that situation, the ability to be upright is a huge improvement if they're unable to walk before. We've mentioned, the idea of Winthrop. We've mentioned the idea of, the applications for this research. Why is it important that this research is going on here? Not at Winthrop, but in this community?
Specifically in the Rock Hill community, there's a large elderly population here. Mm-hmm. So knowing that even having this research in the local area, cognitive disorders usually affect the elderly more so than they do the, I guess, younger population. Mm-hmm. And so I guess being in an area full of people who would depend upon the results of this research would encourage them to, I guess, learn more about what's going on in their community.
Is there a possible economic benefit to the area as well? I mean, do you think that there's potentially, businesses that can be spun up out of research like this, for instance?
Yeah, I mean, that was the thing I was going to think about, that basically research enhances or gives opportunities for potential commercial development. If there was something that would come out of the research, potentially a biotech company could develop, or a technology company if it was something, engineering technology or something that would allow nerves to regenerate or something. And so often that research or these companies will develop near where the research is being developed. They also are recruiting people who have the experience to be able to work for them. So students who are able to get this biomedical research experience could find potential jobs in some of these companies. And that would also, of course, want companies to locate to a place where they recruit employees that would have the skills needed.
Yeah.
So it's sort of a circular amplification.
Yeah. I think this area, we win in a lot of different ways. We win on quality of life. We have great weather. We have access to markets. We have access to airports, beaches, mountains. There are a lot of things that attract businesses to this area. One thing that I think is almost criminally overlooked is this, the ability to, create, to really advance technology in a way where there's, huge advancements in medicine and treatments and, potential businesses. And a problem that most areas try to solve is the problem of brain drain. They want to be able to at least retain their educated, trained workforce. They don't want to be draining people with skills out of the area. And this is an example of something that can really attract highly skilled workers to the area in a way that you can't really it can't be done via a policy, really. It has to be done with, boots on the ground research like you two are doing. And that's going on at Winthrop, and also awareness, right? I mean, awareness is a big deal. Obviously, you guys are here. We're talking about. We're going to put this out everywhere that we can, but that doesn't solve the awareness issue. What do the two of you? What is Winthrop? What is the community? What as a whole do we need to do to help raise the level of awareness, to help increase the probability of those types of successes occurring as a result of what you're doing?
And that's, again, another complex question.
Yeah.
And I really, as I say, my expertise is in doing the research, teaching students, mentoring students. I'll be honest, my strength is not in getting the message out.
Yeah.
And so I think it's working with other people who have the ability to get the message out, people who are investors and wanting to, capitalize on potential commercial advancements. Again, that's not my expertise either.
Yeah.
But I say it's going to take a community of people to do it.
Yeah. I think it does. And, I think, as I said, I think it's almost criminal to the degree that it's been overlooked. And I hope that we can help change that a little bit, because we need a spotlight on what's going on at Winthrop. And also, there's a larger question right now about the value and use of a college education, especially at a time when everyone's trying to do everything virtually, right? So, there's an idea that everyone needs to be able to get their education in a virtual world, and that, there may no longer be a place or as much value in an in-person brick and mortar traditional education. But one of the counter arguments to that would be what the two of you are working on. You can't conduct research like that virtually. You need the physical environment of a lab and the the work of a team and the collaboration in order to really solve those problems. Is that something that y'all have thought about or been aware of through all this last year with, maybe we'll go to school virtually, maybe we'll be in person. How have those policies and decisions affected you, and how do those effects fit into the larger argument about college and where we're going?
So the interesting thing for me as a teacher, I see my students trying to learn virtually, and I've heard from them and I've seen from them that there is an aspect of interaction, both physically if you're doing lab work, and obviously research is very much so, but even learning to do lab work, it's one thing to understand theoretically this is how CRISPR works. It's very different to think of how would I actually do it and what's all the issues that can come up. There's also the whole idea of interaction. I've students physically interacting with their peers and with the professor makes a big difference. I've seen my students say that they struggle when it's just they're left on their own to try to figure it out. Mm-hmm. I think a college education hopefully prepares students that they would be much better able to figure things out on their own. And so potentially would even prepare them for a virtual education to learn this particular thing or this particular aspect or technique. But at the same time, they need to develop those learning skills that are sometimes very hard to develop on their own. Mm-hmm. And again, as you mentioned, anything that's actual practical hands-on, I would not hire a student to work if they only knew the theoretical thing of a technique. Yeah. I want to see that they've had some hands-on, they've gotten it wrong, and then they correct.
Yeah. Which you're kind of on those front lines.
Definitely. From the student line, as far as the classroom goes, not having that teacher interaction and classroom interaction, there's just something missing. The virtual interaction, I guess the information's all there, but it's not just quite the same. Mm-hmm. So I guess it would be like challenging in today's times to need that, I guess, get that education. But like Dr. Birgbauer says, at this point in my education, I'm confident that I can pick up neuroscience papers and gain some understanding about that. And I know I wouldn't be able to do that without him having guided me, or the rest of the teachers at Winthrop having guided me to get the skills required to do that. And so it's definitely important to try to seek that education. Mm-hmm. And as far as lab stuff goes, as if I didn't make a mistake or mess up, to remember to learn that, I didn't do this or I didn't do that, and I was trying to go through it by myself without even someone there to say, hey, you messed that up or you completely got to redo this or something like that, then the advance would slow down. Yeah. Eventually, I'd figure out, this isn't working. I must have did something wrong. Go back and redo it again. But I have someone there to teach me the mistakes that I may make, the potential mistakes that I may make, the things that they've encountered. And so I can grow, I guess, myself to make that scientific advancement or make that scientific discovery or just learn more about a certain aspect to inform others about the certain aspect faster.
And I think you're touching on something, because there's the individual costs to your own education, but I'm almost afraid to even attempt to try to quantify what the collective costs are at the macro level, and the costs of the missed opportunities and the costs of slowing advances and the cost of the research not being done at the same pace because the students that aren't there with the teachers in the lab to actually be able to get things done. I mean, I don't even know how you could quantify that. You're talking about, changing the course of history potentially. So, and what's the cost of that? I mean, that's just unbelievable to think about. But I'm glad that there's still that y'all have figured out a way, and that the school allows you to figure out a way to continue to do research in the physical environment, despite the restrictions. What else? What you said earlier, like, maybe your best skill is not getting the word out. What's one thing or what's something else that you want people to understand? You have a microphone in front of you. Now's your chance. Like, what do you think?
I mean, I guess one thing that I think is really good about Winthrop is that undergraduate students actually get to participate in the research experience, because it's very different to see it by reading it in the textbook where, okay, it's like this is how it works, etc. Then the reality of science that you go in, first of all, we don't quite understand how it works. Even the textbook example is sort of simplified.
Yeah.
And then to realize how did we get there, and how do we get from there to the next stage of understanding and potential application. And that's an experience that is not easy because you don't know the answers yet. You can't read the textbook of what to do next.
Yeah. You have to figure out what to do next.
Yes.
What's the next step, and how does that advance? How does that move the ball?
Yeah.
Were you one of those students in undergrad that was working in the lab? You mentioned you always had a few. Were you one of those when you were in undergrad?
I was actually did get to work with Dr. Birgbauer in my last year of undergrad, and I fell in love with the research.
Yeah.
It made me want to come back and do more.
I was just going to ask you, why did you decide to come back to Winthrop for your master's? Was that why?
It was. I essentially wanted to go, or I still do want to go to med school, but originally research wasn't ever a focus. And being able to do research on neurobiology with Dr. Birgbauer my last year really sparked a passion for research. I found that I enjoyed it, the process of it, the amount that I was exposed to. I did a process called Clarity, and it was using certain bioactive chemicals. So I had biochemistry mixed in with neurology, mixed in with biology, all of that. All of those different aspects of science that I had to juggle and somewhat mastered to accomplish my goal. I enjoyed that a lot.
So you just mentioned potentially med school. Where do you see yourself in 5 years? Do you want to go the PhD route, the MD route? What do you think is your next step?
I want to do...
We're putting them on the spot.
So I've been thinking about a PhD, MD route, but I don't know if I want to be 35 when I'm just starting my career, just getting out of school. So I'm definitely still thinking about the MD route.
Yeah.
But I want to more so concentrate in neurology.
Well, I definitely understand the, not wanting to start at 35. However, I can tell you from the outside, there is no, you shouldn't discount your academic career because you're making great strides. So it's not like you're spinning your wheels until you're 35 waiting to figure out what you want to do. So there's definitely something to be said for what it is you're doing in the meantime. So, but that's interesting. And it's cool to know that, there's a route for someone that can go any direction they want to go, but they can start here at Winthrop and do great things in the meantime. And potentially solve some of the world's great problems. And I think that's pretty cool. But I don't want to keep y'all too long. Thank you so much. This was really exciting. I've been looking forward to this for, 2 weeks now, Trent, since Trent's first set this up for us. But I'm grateful for y'all for coming in today. And I look forward to following up with both of you and hearing in the near future about, further advances and where we are and what's next for you and for Winthrop. So, yeah, thank you so much for coming in today.
Thank you for having us.
Thank you.
Yeah, thanks.